An alternative pattern of growth by cell division, geometric increase, results if all cells of the organism or tissue are active mitotically. Again, starting with a single cell, after round 1 of cell division, there are 2 cells capable of cell division. After round 2, there are 4, and after round 3, there are 8, and so on. The number of cells increases extremely rapidly and can be calculated as a power of 2. After round 3, there are 2 is equal to 8 cells, and after round 10, there are 210 is equal to 1024 cells. After round 20, there are already 220 is equal to 1,048,576 cells. If, as in our previous example, each round of cell division requires 1 day, it now takes the plant only 20 days, not thousands of years, to reach a size of 1 million cells. With geometric increase, a large plant or animal body can be produced quickly. In fact, geometric growth is common in animals but rarely occurs in plants except when they are extremely young and small. The problem is that in plants, dividing cells are typically simple and do not carry out many of the specialized metabolic processes necessary for plant survival. They cannot be the tough, hard fiber cells that make wood strong enough to hold up a tree. They cannot be the waxy, waterproofing cells of the plant's skin that protect it from fungi, bacteria, and loss of water. Plants grow by a combination of arithmetic and geometric increase. A young, embryonic plant grows geometrically and rapidly, with all of its cells dividing. Then cell division becomes restricted to a small group of cells at the tips of roots and shoots. After this point, growth is of the slower arithmetic type. Half of the new cells produced in each round of division remain capable of division, and the other half of the new cells develop into their mature condition and begin carrying out specialized types of metabolism. Plants are thus a mixture of older, mature cells and young, dividing cells. Figure B4, 1 possible types of cell division. Figure 4:18 cytokinesis begins as dictyosome vesicles are trapped by phragmoplast microtubules in the space between the 2 new nuclei. The small vesicles fuse into one large vesicle in which the new middle lamella and 2 primary walls will form. Plasmodesmata form at this time. The cell plate enlarges toward the existing cell wall as more dictyosome vesicles fuse with the edges of the cell plate vesicle. When the cell plate reaches the existing cell walls, the vesicle membrane fuses with the plasma membrane and thus becomes plasma membrane itself. The new cell plate abuts the old cell wall, and the two become glued together with hemocelluloses and pectins. Many meristematic cells are small and cytoplasmic with only a few small vacuoles, but some highly vacuolate cells also divide, for example, vascular cambium cells and large cortex cells forming scar tissue over a wound. In these, the vacuole itself must be divided before either the nucleus or the cell can divide. Vacuole division is accomplished with a phragmosome, a set of microtubules, actin filaments, and cytoplasm. First, the microtubules and actin filaments pull the nucleus to the site where it will divide either along the edge of the cell or often suspended in a tube of cytoplasm that extends through the vacuole. The phragmosome then enlarges as a thin plate-like sheet of cytoplasm and cytoskeleton, extending outward from the vicinity of the nucleus and pushing into the vacuole, dividing it in two. As the phragmosome advances through the vacuole, the nucleus divides, the phragmoplast and cell plate form, and then they too extend outward through the thin sheet of cytoplasm established by the phragmosome. Just after the phragmosome reaches the far side of the vacuole, the phragmoplast and cell plate pass through and join with the plasma membrane and cell wall. The cell has been divided. Meiosis. In mitosis, daughter nuclei are replicates of the original mother nucleus. This is necessary for the growth of an organism but creates a problem when sexual reproduction occurs. 2 sex cells fuse together, forming a zygote, which then grows into a new adult. Each gamete contains one complete set of chromosomes. Nuclei, cells, and organisms with one set of chromosomes in each nucleus are said to be haploid. The zygote has two complete sets, so it is diploid. In most species, the zygote grows into an adult by cell divisions in which the nucleus divides by mitosis. As a result, all cells of the adult are diploid because all nuclei are replicates of the mother nucleus. Figure 4, 19 sperm cells and egg cells are haploid, each having just one set of chromosomes. In this case, the set contains a long chromosome and a short one. After fertilization, the zygote is diploid with two complete sets, two long chromosomes and two short ones. The zygote grows into a mature plant by mitosis, so all nuclei are duplicates of the original zygote nucleus. Each is diploid, with one set each of paternal and maternal chromosomes. Figure 4, 20 meiotic nuclear division consists of two divisions without an intervening S phase. Details are given in text. If the adult were to produce gametes by mitosis, the gametes would be diploid as well, and the next zygote would be tetraploid with 4 sets. It is not possible biologically to double the number of sets per nucleus with each new generation. Instead, it is necessary for a reduction division, called meiosis, to occur somewhere. Meiosis involves 2 rounds of division without allowing the S phase to occur after the first division. The 2 divisions are called meiosis 1 and meiosis 2, and each contains 4 phases similar to those of mitosis. Meiosis occurs only in the production of reproductive cells, gametes in animals and some algae and fungi and spores in plants and other algae and fungi. In seed plants, meiosis occurs in only a few cells in the stamens and ovaries. Meiosis is never used in the growth of the body of any organism. Meiosis 1 prophase I. All events that characterize prophase of mitosis also occur in prophase I. Nucleolus and nuclear membrane break down, centrioles separate, a spindle forms, microtubules attach to centromeres, and chromosomes condense and become visible. In addition, special interactions of chromosomes occur that are unique to prophase I as opposed to prophase of mitosis. Because of this, prophase I is divided into 5 stages. 1. In leptotene, chromosomes begin to condense and become distinguishable, although they appear indistinct. 2. During zygotene, a remarkable pairing of chromosomes occurs. There are 2 sets of chromosomes, one from the paternal and one from the maternal gamete. Because these have gone through S phase in the preceding interphase, each chromosome has 2 chromatids. Altogether there are 4 sets of genes on 2 sets of chromosomes. During zygotene, each chromosome of one set finds and pairs with the equivalent chromosome of the other set. This pairing is synapsis. With remarkable accuracy, the 2 homologous chromosomes in each pair become almost perfectly aligned from end to end. A structure, the synaptonemal complex, is present between the paired homologous chromosomes. This complex is composed of a linear central protein element connected by fine transverse fibers to 2 lateral elements bound to the DNA of the homologous chromosomes. A synapsed pair of homologous chromosomes is called a bivalent. 3. As chromosomes continue to condense, they become shorter and thicker. This stage is pachytene. The synaptonemal complex seems to be involved in the crossing over that occurs now. In several places in each chromosome, the DNA of each homolog breaks. Breaks occur in almost identical places in the paired homologs, and the enzymes that repair the breaks hook the wrong pieces together. A piece of the maternal homolog is attached to the paternal homolog, and the equivalent piece of the paternal homolog is attached to the maternal one. The full consequences of this are explained in texts on genetics and evolution. If the maternal and paternal chromosomes are absolutely identical, nothing significant has happened, but if the genes on the paternal and maternal chromosomes are slightly different, the new chromosomes that result from synapsis and crossing over are slightly different from the original chromosomes. There are no new genes, but rather, there are new combinations of genes on each chromatid. This greatly increases the genetic diversity of all haploid cells produced. Every sperm cell or egg cell produced by any single plant or animal is unique. This genetic diversity is important for evolution. Figure 4.21 During prophase I of meiosis, each paternal chromosome somehow finds in pairs with the equivalent maternal chromosome. They lie parallel to each other. And breaks occur in similar sites on equivalent chromatids, and repair enzymes attach maternal pieces to paternal pieces, resulting in new chromatids. The synaptonemal complex between paired homologous chromosomes. It is not known how it facilitates crossing over. 4. After pachytene is diplotyne. The homologous chromosomes of each bivalent begin to move away from each other but do not separate completely because they are held together at their paired centromeres and at points where they appear to be tangled together. Some biologists believe that chiasmata are the points at which crossing over occurred, but evidence is accumulating that chiasmata are only tangles and are not related to crossing over at all. Under good conditions, it is possible to see all 4 chromatids of the paired homologous chromosomes. They are called tetrads at this stage. 5. In the final stage, diakinesis, homologs continue to separate, and chiasmata are pushed to the ends of the chromosome. The homologous chromosomes become untangled and are paired only at the centromeres. Prophase I is the most complicated stage of meiosis. The remaining stages are simple and quite similar to the stages of mitosis. Metaphase I. Spindle microtubules move the tetrads to the center of the cell, forming a metaphase plate. Anaphase I. The homologous chromosomes separate completely from each other, moving to opposite ends of the spindle. Cohesin is not digested, thus, centromeres do not divide, and each chromosome continues to consist of 2 chromatids. Notice how this is different from the metaphase-anaphase transition of mitosis. In mitosis, separase degrades cohesin, releasing centromeres from each other, and each chromosome divides into 2 chromosomes, each with just 1 chromatid. However, in the metaphase I, anaphase I transition, homologous chromosomes separate from each other, and each still has 2 chromatids. One set of chromosomes is pulled away from the other set, and 2 new nuclei are formed. These nuclei are now haploid because each has only one set of chromosomes. The homologue of each chromosome in one nucleus is now in the other nucleus. Note that each chromosome in anaphase I has 2 chromatids, thus each nucleus has 2 sets of genes, but this does not make them diploid. Any cell in G has at least 2 sets of genes. The critical factor is to have 2 sets of chromosomes. Telophase I. Because the chromosomes are still doubled and are in a G state, they do not need to undergo an interphase with its G, S, and G. Also, because telophase I is basically the opposite of prophase II, some organisms go directly from anaphase I to metaphase II, skipping telophase I and prophase II. In most organisms, however, there is at least a partial telophase I in which chromosomes start to uncoil, and the nucleolus and nuclear envelope start to reappear. If the cells actually progress fully to interphase, no replication of the DNA occurs. The S phase is completely missing. This interval is called interkinesis. Cytokinesis may occur, but it is not unusual for this to be absent also and for both daughter nuclei to stay in the original, undivided mother cell. Meiosis II. If a telophase I occurs, then prophase II is necessary to prepare the nucleus for division. Prophase II is not subdivided into stages like prophase I. Metaphase II is short, and at the end of it, the centromeres divide, thereby separating each chromosome into 2 chromosomes, just as in metaphase of mitosis, but different from metaphase I. Anaphase II then separates each new chromosome from its replicate, and in telophase II, new nuclei are formed. Each nucleus contains just one set of chromosomes, each with a single chromatid. To summarize, during meiosis I, each chromosome of the paternal chromosomes undergoes synapsis with the homologous chromosome of the maternal set. Crossing over results in new combinations of genes on the chromosomes. Spindle microtubules pull the paired homologs to opposite poles, therefore, at each pole, there is only one set of chromosomes, not two. The new nuclei that form temporarily after meiosis I are thus haploid. When these nuclei undergo meiosis II, the two chromatids of each chromosome are separated, as in mitosis, and the resulting nuclei each have a haploid set of chromosomes, each chromosome with only one chromatid. In meiosis, nuclear division and cell division are often not directly linked. In some species, cytokinesis happens after both meiosis I and meiosis II, and 4 haploid cells result from each original diploid mother cell. In other species, no cytokinesis occurs after meiosis I, but a double cytokinesis occurs after meiosis II, again resulting in 4 haploid cells. For example, pollen grains can form either way, and meiosis results in 4 haploid cells. However, in many organisms, no cytokinesis occurs at all during the meiosis that leads to the formation of eggs. The final cell is tetranucleate and may remain that way, or 3 of the nuclei may degenerate and produce only a single uninucleate haploid cell by meiosis. It seems to be selectively advantageous to produce one large cell rather than 4 small ones. Figure 4.22 In animals and some plants, the meiosis that is part of egg production produces only one large haploid cell. In some species, meiosis results in 4 haploid nuclei, no cytokinesis occurs and all 4 nuclei are temporarily in one cell until degeneration eliminates 3 of them. In other species, after meiosis I occurs, cytokinesis places one nucleus in a small cell, and both nucleus and cell degenerate before they can undergo meiosis II. The nucleus of the larger cell does undergo meiosis II, and again, one daughter nucleus is placed in a small cell that degenerates. Only 3 nuclei are ever formed, and only one survives. Less common types of division in plants. Cytokinesis and karyokinesis are often so closely associated that we tend to think of them as 2 aspects of one process. In numerous tissues, however, karyokinesis occurs without cytokinesis, and multinucleate cells are formed. The nutritive tissue of many seeds goes through a phase in which its cells are multinucleate, and in many algae, all cells are like this. If the cell becomes very large and has hundreds or thousands of nuclei, it is called a koinocyte. Figure 4.23 A multinucleate cell in the developing seeds of Capsella. Although numerous nuclei have undergone mitotic divisions, the cell has not divided but has enlarged and now has a giant vacuole. The small ball of cells is the embryo, which pushes into the multinucleate koinocyte and draws nutrients from it. Multinucleate cells are typical of many algae. This is Valonia. The entire alga pictured here consists of just 3 giant cells, but each has thousands of nuclei. A magnification of, showing some of the many nuclei, which have been stained pink. On the other hand, cell division may occur without nuclear division. This is most common in algae, fungi, and the nutritive tissues of seeds. The cells first become multinucleate and persist in that condition for some time, then cell division begins as new walls are organized around each nucleus and its accompanying cytoplasm. In lily flowers, each haploid cell involved in producing an egg cell undergoes 2 rounds of mitosis without cytokinesis, resulting in a tetranucleate cell. 3 nuclei migrate to one end of the cell, the 4th nucleus migrates to the other, then all 4 undergo mitosis again. The isolated nucleus divides into 2 nuclei as would be expected. However, as the 3 crowded nuclei begin to divide, their spindles fuse into a single structure. Thus, all 3 sets of chromatids on one side go to one pole of the spindle, and all 3 on the other side go to the other spindle. The result is not 6 haploid nuclei but instead 2 triploid nuclei. The cell now has 2 small haploid nuclei and 2 large triploid nuclei. The egg is always formed from one of the haploid nuclei, never one of the triploid ones. Figure 4.24 As part of egg production in lilies, certain cells go through an odd karyokinesis in which 3 haploid nuclei divide, but because they lie so close to each other, their spindles fuse and 2 new triploid nuclei are formed instead of 6 haploid ones. Cell division in algae. Nuclei. The nuclei of plants are virtually identical to those of animals in terms of structure, metabolism, mitosis, and meiosis. Apparently, the plant and animal lines of evolution diverged only after the eukaryotic nucleus had become quite sophisticated. However, nuclei are complex organelles, and mitosis and meiosis are intricate processes with many steps. Certainly plant and animal nuclei could not have arisen from prokaryotic nucleoids quickly. Many intermediate steps spanning hundreds of millions of years must have been involved. Several groups of organisms with unusual nuclear characteristics may represent lines of evolution that originated earlier in the history of eukaryotes than did plants and animals. Nuclei of many fungi have an unusual mitosis, some having both an intranuclear and an extranuclear spindle. In many fungal species, the nuclear envelope and nucleolus either do not break down at all or do so only late in mitosis. Similar behavior occurs in many groups of algae, including dinoflagellates, brown algae, euglenoids, and some green algae. Gaps form in the nuclear envelope, and bundles of microtubules pass completely through the nucleus. Even more significantly, nuclei of dinoflagellates have no histones. Their chromosomes remain condensed at all times, undergoing only a slight uncoiling during interphase. Because histone and nucleosome structure is so constant in all other eukaryotes, this unique situation in dinoflagellates is considered highly significant. Figure 4:25 late telophase in the green alga Cladophora. Not only is the nuclear membrane completely intact, but a portion of the intranuclear spindle is visible. An interphase nucleus of dinoflagellate. Chromosomes never completely decondense. It is not known how chemical messengers, DNA replicases, and RNA synthetases gain access to the DNA molecule. Cytokinesis. Several types of cytokinesis occur in algae. Many groups of unicellular algae have no wall, their plasma membrane being their outermost surface. During cell division, the plasma membrane pinches in two, being pulled inward as a cleavage furrow, a process remarkably similar to cytokinesis in animals. In many algae with walls, cell division is similar to that of plants. After telophase, a phragmoplast forms, and dictyosome vesicles establish a cell plate, which then grows radially outward, centrifugal growth, until it meets and fuses with the wall of the parental cell. In some green algae, cytokinesis occurs by a different method. The mitotic spindle depolymerizes quickly, and the two daughter nuclei lie close together. A new set of microtubules appears between them, oriented parallel to the plane where the new wall will form, which is perpendicular to the orientation of the spindle. This group of microtubules is a phycoplast and may be associated with division either by furrowing or by cell plate formation. Phycoplasts are associated with those green algae in which the nuclear envelope does not break down during mitosis, whereas phragmoplasts are associated with the loss of the nuclear envelope. Plants arose from green algae that divide with a phragmoplast rather than a phycoplast. Figure 4:26 in unicellular, wall-less forms, cell division occurs by unfurrowing of the plasma membrane. Most algae with walls divide by a phragmoplast and cell plate, just as plant cells do. And some green algae have a phycoplast. The mitotic spindle disorganizes quickly, after which a new set of microtubules polymerizes perpendicular to the spindle axis. Division may then occur by cell plate growth or by unfurrowing. In red algae, cell division occurs by a phragmoplast, but the new wall grows inward from the pre-existing walls. It stops growing while still incomplete, having a large hole called a pit connection, which is filled with protein and carbohydrate material. Otherwise, most aspects of wall formation are similar to those of other walled algae. Figure 4:27 cross walls of red algae are unusual in being incomplete, having a hole in their center. This is closed by a pit plug. Cynia confusa. Cell division of prokaryotes. The events of mitosis and meiosis occur only in eukaryotes. In prokaryotes, bacteria, cyanobacteria, and archaea, mitosis and meiosis do not occur, and cytokinesis is much simpler. All genetic material occurs as one or several rings of DNA, which does not have any protective histones. The loops of DNA are attached to the cell's plasma membrane, and when the DNA is replicated, the two daughter loops also are both attached to the membrane. Because no centromere holds them together, the circles of DNA are pulled apart as the cell and its membranes grow. Several new rounds of replication may occur before cell division takes place, and thus each cell may have as many as 20 identical circles. Cytokinesis occurs by a process of unfurrowing. The plasma membrane pulls inward and finally pinches in two. As the plasma membrane furrows inward, a new cross-wall grows inward, starting from the existing wall. After completion, the cross-wall splits, becoming two walls and releasing the two daughter cells. Multicellular aggregates are rarely formed in bacteria and archaea. In cyanobacteria, cross-walls do not break apart, and multicellular bodies are formed. The cell cycle of many bacteria can be short, only 20 minutes under ideal conditions. For many species, however, the cycle may last several days or weeks even under optimal conditions. Figure 4.28 In prokaryotes, the circles of DNA are attached directly to the plasma membrane. As the cell and its membrane grow, the attached circles of DNA are separated. Cell division then occurs between the circles. Botany and beyond. Box 4-3 Chloroplast division during leaf growth. Elegant studies have been done of the growth and division of plastids in relationship to growth and development of leaves in spinach. In very small leaves, 1 mm long or less, plastid DNA constitutes 7% of the total cell DNA, and an average of 76 DNA circles are present in each plastid. As the leaf doubles in size to 2 mm long, plastid DNA is replicated at approximately the same rate as nuclear DNA, and thus, it remains low, approximately 8% of total cellular DNA. As the leaf continues to expand to 20 mm long, plastid DNA is replicated much more rapidly than nuclear DNA and increases to 23% of total cellular DNA. At the same time, the number of plastids per cell triples from 10 to 29, and thus, neither plastid division nor plastid DNA replication is controlled by the mechanisms that govern cell or nuclear DNA replication. At this point, each plastid has 190 DNA circles, and each cell has a total of 5,510. In the next stage of leaf growth, to 100 mm long, no synthesis of DNA occurs, and no new cells form. Instead, those already present expand. However, plastids continue to divide even though they are not making any more DNA. Consequently, the number of DNA circles per plastid drops from 190 to 32, whereas the number of plastids per cell increases from 29 to 171. From these data, it is reasonable to form the hypothesis that plastid growth, DNA replication, division, and development are correlated predominantly with tissue or organ development rather than with the cell cycle. Table B4.3 Development of the plastid genome in spinach leaves. Data from Scott, N.S., and J. V. Possingham, 1983. Changes in chloroplast DNA levels during growth of spinach leaves. J. Experimental Bot 34:1756, 67. Division of chloroplasts and mitochondria. Mitochondria and plastids are constructed similarly to prokaryotes. They also contain circles of naked DNA that become separated by membrane growth. Division of the organelles is accomplished either by unfurrowing or by being pulled in two. Because they contain DNA necessary for their growth and functioning, each daughter cell must receive at least one mitochondrion and one plastid during cytokinesis. If not, the cell lacks that organellar portion of its genome and cannot produce the organelle. This happens occasionally with plastids and is often not a serious problem. The cell survives by importing sugar from neighboring cells and can grow along with the tissue. All daughter cells also lack plastids, so if this occurs in a young leaf, a white spot forms. The same phenomenon probably occurs with mitochondria but is more difficult to detect. The metabolic stimulus that triggers the replication of nuclear DNA is not the stimulus that controls replication of organellar DNA. During the cell cycle, replication of nuclear DNA is episodic, occurring as discrete episodes that occupy a small portion of the total cell cycle, S phase. However, the duplication of the rest of the cells seems to be continuous. The volumes of plastids, mitochondria, cytosol, endoplasmic reticulum, and other organelles appear to increase gradually and steadily throughout interphase rather than in discrete episodes. Figure 4, 29 micrograph of a chloroplast dividing in Arabidopsis thaliana. As the chloroplast divides, its central region is pinched inward by a contracting ring of proteins, which constricts the chloroplast into a dumbbell shape. The chloroplast shines brightly because of the way its chlorophyll reacts to the light used by the microscope. It absorbs the light and re-emits it as a different color. At the next level. 1. Orientation of cell division. The orientation of cell division in plants determines the positions of the cells as they differentiate. Plant cells cannot migrate to new positions in the way animal cells can. They must be produced in the proper place. Cells usually divide into two cells that are more or less equal in size, but during the formation of stomata in the epidermis, cells often divide asymmetrically into a large cell and a small one. Also, when a plant is wounded, surviving cells near the wound typically divide in such a way as to produce a layer of protective scar cells near the wound. The mechanisms that determine where the pre-prophase band forms are critical in establishing the plane of cell division. 2. Control of the cell cycle. Chemical factors control when cells replicate their DNA, when they start to divide, how rapidly they divide, and when they stop. Cells and growing tissues divide repeatedly, but at some point, some or all these cells must stop dividing and begin to differentiate into specialized, mature cells. Also during autumn or when stressed, cell division must be slowed or stopped, whereas during seed germination, certain factors influence certain cells to initiate the division cycle. Another important but poorly understood process determines whether cells undergo mitosis or meiosis. 3. Cell formation without nuclear division. During seed development in angiosperms, a tissue called endosperm develops in an unusual manner. In the first stage, endosperm nuclei divide frequently but there is no cell division, so a giant multinucleate cell is formed. The center of an unripe coconut is one of these giant cells. The coconut milk is the cell's protoplasm. In the second stage, hundreds of cell walls form, trapping some cytoplasm and nuclei, creating hundreds or thousands of cells. This is cellularization of endosperm and it produces the white coconut meat we use in candy, cake, and pies. Summary. 1. Cells have a life cycle, the cell cycle, consisting of alternating phases of growth and division. In organs that live just briefly, cells cycle only while the tissue is young and then enlarge and differentiate. 2. The interphase portion of the cell cycle consists of G, S, and G. During G, the nucleus controls cell metabolism. During S, the DNA is replicated. In G, the cell prepares for division. 3. Each DNA molecule contains thousands of regions, genes, that contain the information for proteins. A chromosome is a complex in which histone proteins bind to DNA and stabilize it. Each chromosome consists of a centromere and either one chromatid before S phase or two chromatids after S phase. Chromatid ends are telomeres. 4. Cell division is cytokinesis, and nuclear division is karyokinesis. The words mitosis and meiosis technically refer to only karyokinesis. 5. During mitosis, spindle microtubules pull the two chromatids of each replicated chromosome to opposite ends of the cell. Because the two chromatids are virtually identical, the two new nuclei are also almost identical to each other. 6. Nuclei of any ploidy level, haploid, diploid, and higher ploidies, can undergo mitosis. During growth of the plant body, All nuclear divisions are mitotic divisions. 7. Meiosis is reduction division. Diploid cells produce haploid cells. Just as important, genetic diversity is increased by meiosis. 8. During prophase I, homologous chromosomes pair and exchange pieces of chromatid. At the end of prophase I, the nucleus no longer has purely paternal and purely maternal chromosomes. During anaphase I, homologous chromosomes are pulled away from each other, reducing the number of sets of chromosomes to half the original number. Haploid, triploid, and all other oddploid nuclei cannot undergo meiosis. 9. Meiosis occurs only in the production of reproductive cells, either gametes or spores. Meiosis is never used in the growth of the body of any organism. 10. Cytokinesis in plants and algae occurs by the formation of a large vesicle between the forming daughter nuclei. Within the vesicle, the middle lamella and two new walls form and expand until they reach existing walls. 11. Cytokinesis and karyokinesis are usually closely coordinated, but nuclear division can occur without cell division, resulting in multinucleate cells. Important terms. Anaphase arithmetic increase cell cycle cell plate centromere chromatid chromosome. Chromosome condensation crossing over cytokinesis diploid. Duplication division G-phase gametes genome geometric increase haploid histones. Homologous chromosome interphase karyokinesis kinetochore meiosis metaphase plate mitosis phragmoplast. Phragmosome phycoplast pre-prophase band prophase reduction division resting phase S phase spindle. Spindle microtubule synapsis telophase tetraploid zygote. Review questions. 1. In a woody plant such as a tree, which parts have cells that live only briefly and die quickly? Which parts have cells that live for several years? 2. Some cells never stop dividing. Give 2 examples of cells like this. 3. Interphase is also called the resting phase of the cell cycle. Why was it given that name? 4. What are the main activities of a cell while it is in G phase? What kinds of organisms have short G phases, and how long does a short G phase last? What kinds of organisms have long G phases, and how long do they last? 5. What is the main activity of the S phase of the cell cycle? What does S stand for? 6. Table 4. 2 gives the number of chromosomes in a haploid set of chromosomes. What is the lowest number in the table? What is the highest number? Most cells in plants are diploid, having 2 sets of chromosomes, so the number per nucleus should be doubled. How many chromosomes are present in each diploid potato nucleus? If a potato plant has 1 million cells, each with 1 diploid nucleus, how many chromosomes are present in the entire plant? 7. How many chromatids does a chromosome have before S phase of the cell cycle? How many does it have after S phase? 8. Examine Table 4. 1. How many hours does the cell cycle last in the root tips of corn and an onion? Which plant has cells that divide more quickly? How many cell cycles could corn and onion roots undergo in 4 weeks? If after a cell divides both daughter cells could divide and then their daughter cells could divide? How many corn cells and how many onion cells would be present at the end of 4 weeks if you started with just 1 cell of each? 9. What are the 4 phases of the cell cycle? What is the principal activity in the cell during each phase? Can any phase be eliminated or bypassed? 10. Why is mitosis called duplication division and meiosis called reduction division? What is reduced and what is duplicated, chromosomes, number of chromosomes, or number of sets of chromosomes? 11. What does it mean when chromosomes are said to condense during prophase of mitosis? How long are chromosomes after condensation is complete? How big is a typical dividing cell in a root or shoot? If a chromosome were still 20 micrometers long after condensation, would it be possible for division to pull half of it to one end of the cell and the other half to the other end? 12. What is the name of the set of microtubules that pull chromosomes apart? What is the name of the attachment point between microtubules and chromosomes? What is the name of the ends of the cells where the chromosomes are pulled? 13. What is a metaphase plate? What is duplicated at the end of metaphase? When this is duplicated, chromatids become free of each other. How many chromatids are there per chromosome in prophase? How many chromatids per chromosome after metaphase? 14. Imagine a nucleus that has 10 chromosomes. How many chromosomes does it have before prophase begins? How many chromosomes does each daughter nucleus have after telophase has been completed? How many chromatids are present in the one mother nucleus before prophase begins? How many chromatids are present in each daughter nucleus after telophase has been completed? How many chromatids are present? If you add together all chromatids in both daughter nuclei after telophase has been completed? 15. What are the 4 phases of mitosis, and what is the principal activity in the nucleus during each phase? 16. Draw a single, imaginary chromosome as it would appear just as mitosis is ending. Now describe what happens to it during interphase and then during mitosis. Be especially careful to consider how many chromatids and how many copies of each gene it has at each stage. 17. How does cytokinesis occur in plants? Which organelle produces vesicles that fuse to form the cell plate? What membrane is transformed into new plasma membrane? 18. Many people consider algae to be plants even though algae do not have roots, stems, and leaves, but in many algae, cell division is different from that in true plants. Do most algae have a phragmoplast? What is the name of the structure they use? 19. Sex cells are also called. If a species has males and females, males produce sex cells called cells, and females produce sex cells called cells. 20. How many sets of chromosomes does each sperm cell carry? How many does each egg cell have? Gametes such as sperm cells and egg cells are said to be. After a sperm and an egg have fused, the new cell is called a. And it is. 21. If a plant has diploid cells with 20 chromosomes in each nucleus, how many chromosomes does each of its gametes have? What is the name of the division that reduces the number of chromosomes from 20? 22. What are the 5 stages of prophase I, and what is the principal activity of the nucleus and chromosomes during each stage? 23. During zygotine stage of meiosis I, a remarkable pairing of chromosomes occurs. Look at the bottom part of figure 4:19 in which mitosis, growth of the body, has produced cells with 4 different types of chromosomes. The black ones came from the sperm nucleus. The red ones came from the egg nucleus. If one of these nuclei were to undergo meiosis, which chromosomes would pair during zygotine, the long black one with the short black one or the long black one with the long red one? 24. What is the name of the structure that holds homologous chromosomes together after they have paired during zygotine? 25. During pachytene, crossing over occurs. Do chromosomes actually break during this process? 26. How does anaphase I of meiosis differ from anaphase of mitosis? Does the number of chromatids per chromosome change in anaphase I? Does the number of sets of chromosomes per nucleus change in anaphase I? 27. Draw all stages in the cell cycle and meiosis for a nucleus that has just one pair of homologous chromosomes, and then do the same for a nucleus that has 3 different types of chromosomes. Draw all stages. 28. If a cell undergoes nuclear division but not cell division, it becomes multinucleate. If it has hundreds of nuclei, it is called a. 29. Do prokaryotes undergo meiosis or mitosis? How do their cells divide? 30. Describe the process of division that produces more plastids and mitochondria. Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated ZArt. Chapter 5. Tissues and the primary growth of stems. Chapter opener image. These strange little plants are seedlings of a desert-adapted species, Hoodia gordonii. Even at this young stage, each has most of the basic plant tissues that protect the body, conduct materials through it, carry out photosynthesis, and so on. At the appropriate time, each will also make wood and bark, and also flowers, fruits, and seeds. These seedlings are being cultivated in a greenhouse, but the species is native to deserts in Southwest Africa. Outline. Concepts. Basic types of cells and tissues. Parenchyma, Collenchyma, Sclerenchyma. External organization of stems. Internal organization of stems, arrangement of primary tissues. Epidermis, cortex, vascular tissues, xylem, phloem, vascular bundles. Stem growth and differentiation. Box 5 to 1 plants and people, parenchyma, sclerenchyma, and food. Box 5 to 2 plants do things differently, organs, replace them or reuse them. Box 5 to 3 alternatives, simple plants. Box 5 to 4 plants do things differently, plants and people grow differently. Learning objectives. After reading this chapter, students will be able to summarize the evolution of land plants. Give examples of herbs and woody plants. Define the 3 classes of plant cells. Describe the components of basic stem organization. Provide examples of modifications to basic stem components. Recall the components of the internal organization of stems. State the functions of the epidermis. Contrast the way water moves through tracheids as compared to vessel elements. Order the processes of primary growth. Did you know? All soft parts of plants, such as flowers and leaves, are composed of cells that live only briefly, just a few days for flowers, just from spring until autumn for most leaves. Most hard parts of plants, such as wood and the shells around seeds, are composed of cells that usually die immediately after they have hardened. Almost all shoots, even the highly modified shoots of cacti, orchids, bulbs, and vines, have just one pattern of tissues. Once you have learned the pattern of one stem, you know the pattern present in almost all. Concepts. The body of an herb contains just 3 basic parts: leaves, stems, and roots. When the first land plants evolved about 420 million years ago, they were basically just algae that either washed up onto a shore or were left there as lakes and streams evaporated. They had no roots, stems, or leaves, and they just lay on the mud. As the shores gradually became crowded with such plants, some grew over others, shading them. Any plant that had a mutation allowing it to grow upright into the sunshine above the others had a selective advantage. However, being upright is not easy. Elevated cells are out of contact with the moisture of the mud, therefore, water must be transported up to them. Elevated tissues act as a sail and tend to blow down, so supportive tissue is necessary. Absorptive cells in mud are shaded and cannot photosynthesize, thus, sugars must be transported down to them. Shortly after plants began living on land, distinct, specialized tissues and organs began to evolve. As early populations of land plants continued to evolve and became taller, their stems functioned primarily as transport and support structures, as they do still. But, stems of modern flowering plants have additional roles. They produce leaves and hold them in the sunlight, and during winter, they store sugars and other nutrients, such as the sugary sap of maples. Stems may also be a means of survival. Underground bulbs and corms remain alive when above-ground leaves die. Stems of many species are a means of dispersal. They spread as runners or vines, or pieces break off and are carried by animals or water to new areas where they sprout roots and grow into new plants. Although all flowering plants possess leaves, stems, and roots, these parts have been modified so extensively in some species that they may not be recognizable without careful study. For example, cacti are often described as leafless, but they actually have small green leaves between 100 and 1,000 millimeters long. Large, broad leaves would be selectively disadvantageous for these desert plants because the extensive surface area of such leaves gives up so much water to the dry air that the plant would desiccate. Similarly, all flowering plants have stems, but in some, they are only temporary, reduced structures. Orchids such as Campylocentrum pachyrizum and Heracella perecta consist of a mass of green photosynthetic roots connected to a tiny portion of stem. Roots constitute almost the entire plant body. The shoot becomes active only when flowers are to be produced. One hypothesis is that this unusual body evolved because the ancestors of these species had roots that were more resistant to water stress than their stems were. Such plants could occupy drier habitats if either of two things happened. Mutations were selected that caused stems and leaves to be more water-conserving. This happened in most orchid species, or mutations occurred that enhanced the root's ability to absorb carbon dioxide and carry out photosynthesis. Although thousands of plant species can withstand harsh conditions, only a handful do so by being shootless and having photosynthetic roots. Mutations that permit this type of body may be either rare or generally detrimental. Figure 5.1 The primary body of an herb like this geranium consists of roots, stems, and leaves. Buds are located in the axil of each leaf and may grow to be either a vegetative branch or a set of flowers. This iris is also an herb and never produces wood or bark. Almost all flowering plants are either broadleaf plants like the geranium or plants with grass-like leaves like the iris. Figure 5.2 This prickly pear shows that one plant can have two types of shoot. The pad is the main shoot, and the spine clusters are highly modified axillary branch shoots. One of the spine-bearing branches has been stimulated to develop into the first type of shoot and become a pad-like branch. The plant also has two types of leaves, small fleshy green leaves on the young buds and spines on the axillary branches. Some plants in the bromeliad family are nearly rootless. In the coastal deserts near Lima, Peru, fog is frequent, but rain never falls. Because the soil is always dry, roots are of little use. Plants of Tillandsia straminia of that region are small, herbaceous vines that lie on top of the soil. Plants absorb moisture through leaves made wet by fog, and they derive minerals from wind-blown dust that dissolves on moist leaf surfaces. The plants are not anchored to the soil but roll over coastal dunes as the wind blows them. Such a lifestyle would be impossible for a tree or bush because large plants could not absorb enough water or minerals without roots. Figure 5.3 This orchid carries out photosynthesis by means of roots that contain chlorophyll. The plant grows in trees, attached to their bark such that its roots are exposed to sunlight. They are never in soil. The photosynthetic roots grow in any direction, even upward. The plant has only a small portion of shoot, which produces flowers. Figure 5. For most epiphytic orchids resist the stresses of temporary drying because their shoots are fibrous and have a thick cuticle composed of cutin and wax. In each plant described, leaves, stems, or roots have become highly modified by natural selection, permitting survival in unusual habitats. However, in no species have any of these organs been completely lost evolutionarily. We must assume that the organ carries out some essential function. In shootless orchids, the residual shoot is necessary for flowering and sexual reproduction. Cactus leaves are involved in the formation of buds that produce the defensive spines. Although roots of T. straminia neither absorb water and minerals nor anchor the plant, they may be essential for production of critical hormones, as are roots of other plants. In all cases, analysis of many aspects of the plant's biology reveals how the structure and metabolism of a particular organ are adaptive, how modifications affect other plant parts, and how organs have subtle functions not always obvious in more typical plants. Figure 5. These bromeliads are not rooted into the coastal sand dunes, a strong wind can blow them around. All water is absorbed from fog condensing on the leaves. Table 5. 1. Types of plant body. The flowering plants discussed here are formally classified as the division Magnoliophyta, but they are known informally as angiosperms. This group consists of approximately 297,000 species and is the largest division in the plant kingdom. Early angiosperms diversified into several groups that are now known as basal angiosperms, eudicots, and monocots. Most basal angiosperms probably will not be familiar to you except for water lilies, magnolias, and laurels. Eudicots are broadleaf plants such as roses, asters, maples, and others, and monocots are grasses, lilies, cattails, palms, philodendrons, bromeliads, and several others. This chapter began by stating that the body of an herb contains 3 parts. It is necessary to explain more precisely what an herb is. Plant bodies are of 2 fundamental types, an herbaceous body, also called a primary plant body, and a woody body, known as a secondary plant body. An herb is a plant that never becomes woody and covered with bark. It often lives for less than a single year, although many live and grow for years. Its tissues are primary tissues. In woody plants such as trees and shrubs, the wood and bark are secondary tissues. A few plants are surprising. Palm trees are large, perennial, and very hard, but they do not actually have wood, thus, they are giant herbs, not woody plants. Basic types of cells and tissues. Despite the diversity of types of stems that have originated by natural selection, all share a basic, rather simple organization. The same is true for leaves and roots. Although we might suspect that a plant contains numerous types of cells, actually, all plant cells are customarily grouped into just 3 classes based on the nature of their walls: parenchyma, collenchyma, and sclerenchyma. Parenchyma. Parenchyma cells have only primary walls that remain thin. Parenchyma tissue is a mass of parenchyma cells. This is the most common type of cell and tissue, constituting all soft parts of a plant. Soft leaves, petals, fruits, and seeds are composed almost completely of parenchyma. Parenchyma cells are active metabolically and usually remain alive after they mature. Numerous subtypes are specialized for particular tasks. Chlorenchyma cells are parenchyma cells involved in photosynthesis. They have numerous chloroplasts, and their thin walls allow light and carbon dioxide to pass through to the chloroplasts. Other types of pigmented cells, as in flower petals and fruits, also must be parenchyma cells with thin walls that permit the pigments in the protoplasm to be seen. Glandular cells that secrete nectar, fragrances, mucilage, resins, and oils are also parenchyma cells. They typically contain few chloroplasts but have elevated amounts of dictyosomes and endoplasmic reticulum. They transport large quantities of sugar and minerals into themselves, transform them metabolically, and then transport the product out. Transverse cells are parenchyma cells that mediate short-distance transport of material by means of a large, extensive plasma membrane capable of holding numerous molecular pumps. Unlike animal cells, plant cells cannot form folds or projections of their plasma membranes. Instead, transverse cells increase their surface area by having extensive knobs, ridges, and other ingrowths on the inner surface of their walls. Because the plasma membrane follows the contour of all these, it is extensive and capable of large-scale molecular pumping. Some parenchyma cells function by dying at maturity. Structures such as stamens and some fruits must open and release pollen or seeds. The opening may be formed by parenchyma cells that die and break down or are torn apart. Large spaces are often needed inside the plant body, for example, to allow gases to diffuse within leaves. Many such spaces are formed when the middle lamella decomposes and cells are released from their neighbors. In other cases, the space is formed by the degeneration of parenchyma cells. In a few species, such as milkweeds, as parenchyma cells die, their protoplasm is converted metabolically into mucilage or a milky latex. Table 5, 2-3 basic types of plant cells and tissues, based on cell wall. Figure 5, 6 parenchyma cells of geranium. Their walls are thin, and their vacuoles are large and full of watery contents that did not stain. Nuclei were present in all cells, but because these cells were so large and the section was cut so thin, most nuclei were cut away during the preparation of this slide. One nucleus is still present. Chlorenchyma cells from a leaf of privet. Because these cells are small and the section is thicker than that in, most cells still have nuclei. The structures close to the wall are chloroplasts. The large white areas are intracellular spaces where the cells have pulled away from each other, permitting carbon dioxide to diffuse rapidly throughout the leaf. Material taken from the center of a pine stem. Cells that have stained dark purple are filled with chemicals called tannins. These are bitter and deter insects from eating the tissue. A resin canal in a pine leaf. The white area is the central cavity where resin is stored, and cells that line the cavity are glandular parenchyma cells that synthesize and secrete resin. The innermost cells have thin walls, which permit movement of resin from the cells to the cavity. The outer cells have thick walls, which provide strength. Cells with thick walls are not parenchyma cells. Figure 5, 7 transverse cells in the salt gland of Franknia grandifolia. The wall ingrowths increase the surface area of the cell membrane, providing more room for salt-pumping proteins in the membrane. These transverse cells were frozen, then broken open, and all of the protoplasm was washed away with detergent. Only the cell wall is left. All the knobby irregular areas are wall ingrowths. Parenchyma tissue that conducts nutrients over long distances is phloem. It is discussed later in this chapter. Parenchyma cells are relatively inexpensive to build because little glucose is expended in constructing the cellulose and hemicellulose of such thin walls. Most parenchyma cell walls are only 80 to 100 nanometers thick, with just 5 to 10 layers of cellulose microfibrils. Each molecule incorporated into a wall polymer cannot be used for other functions such as generating ATP or synthesizing proteins. Consequently, it is disadvantageous to use a cell with thick walls any time one with thin walls would be just as functional. Most leaves are soft, composed almost entirely of parenchyma, and are therefore not very expensive metabolically. After several weeks of photosynthesis, they replace the sugar used in their construction, and all photosynthesis after that point is net gain for the plant. Chlorenchyma. Chlorenchyma cells have a primary wall that remains thin in some areas but becomes thickened in other areas, most often in the corners. The nature of this wall is important in understanding why it exists and how it functions in the plant. Like clay, the wall of chlorenchyma exhibits plasticity, the ability to be deformed by pressure or tension and to retain the new shape even if the pressure or tension ceases. Parenchyma is present in elongating shoot tips that must be long and flexible, such as those of vining plants like grapes. It is present as a layer just under the epidermis or as bands located next to vascular bundles, making the tips stronger and more resistant to breaking. However, the tips can still elongate because parenchyma can be stretched. In species whose shoot tips are composed only of weak parenchyma, the tips are flexible and delicate and often can be damaged by wind. The elongating portion must be very short, or it simply buckles under its own weight. It is important to think about the method by which parenchyma provides support. If a vine or other parenchyma-rich tissue is cut off from its water supply, it wilts and droops. The parenchyma is unable to hold up the stem. Parenchyma cells are needed in the inner tissues for support. Parenchyma and turgid parenchyma work together like air pressure and a tire. The tire or inner tube is extremely strong but is useless for support without air pressure. Similarly, air pressure is useless unless it is confined by a container. In stems, the tendency for parenchyma to expand is counterbalanced by the resistance of the parenchyma, and the stem becomes rigid but able to grow. Because parenchyma cell walls are thick, they require more glucose for their production. Parenchyma is usually produced only in shoot tips and young petioles, where the need for extra strength justifies the metabolic cost. Subterranean shoots and roots do not need parenchyma because soil provides support, but aerial roots of epiphytes such as orchids and philodendrons have a thick layer of parenchyma. Sclerenchyma. The third basic type of cell and tissue, sclerenchyma, has both a primary wall and a thick secondary wall that is almost always lignified. These walls are elastic. They can be deformed, but they return to their original size and shape when the pressure or tension is released. Sclerenchyma cells develop from parenchyma cells in mature organs after they have stopped growing and have achieved their proper size and shape. Deforming forces such as wind, animals, or snow are usually detrimental. If mature organs had parenchyma for support, they would be reshaped constantly by storms or animals, which of course would not be optimal. For example, while growing and elongating, a young leaf must be supported by parenchyma if it is to continue to grow, but after it has achieved its mature size and shape, some cells of the leaf can mature into sclerenchyma and provide elastic support that maintains the leaf's shape. Unlike parenchyma, sclerenchyma supports the plant by its strength alone. If sclerenchyma-rich stems are allowed to wilt, they remain upright and do not droop. Figure 5-8 masses of parenchyma cells often occur in the outer parts of stems and leaf stalks. This is part of a peperomious stem. Parenchyma forms a band about 8 to 12 cells thick. The inner part of the stem is mostly parenchyma. In parenchyma cells, the primary wall is thicker at the corners so the protoplast becomes rounded. No intercellular spaces are present. Figure 5-9 shoot tips of long vines need the plastic support of parenchyma while their stems are elongating. Parenchyma and parenchyma cells can absorb water so powerfully that they swell and stretch their walls, thereby growing. Sclerenchyma cell walls are strong enough to prevent the protoplast from expanding. The rigidity of sclerenchyma makes it unusable for growing shoot tips because it would prevent further shoot elongation. Sclerenchyma cells are of two types, conducting sclerenchyma and mechanical sclerenchyma. The latter type is subdivided into long fibers and short sclerids, both of which have elastic secondary walls. Because fibers are long, they are flexible and are most often found in areas where strength and flexibility are important. The wood of most flowering plants contains abundant fibers, and their strength supports the tree while their elasticity allows the trunk and branches to sway in the wind without breaking or becoming permanently bent. Our ribs are similarly strong, flexible, and elastic. Fiber-rich bark resists insects, fungi, and other pests. Table 5, 3 types of sclerenchyma. Figure 5-10 A mass of fiber cells in the leaf of agave. These are large, heavy, tough leaves, and fiber masses provide elastic strength. Each cell consists mostly of thick secondary cell wall. The small white space in each is an area where the protoplast had been before it died. The same mass of fibers as in but viewed with polarized light. Secondary walls shine brightly because their cellulose molecules are packed in a tight, crystalline form, giving the wall extra strength. A stem of bamboo was treated with nitric and chromic acid to dissolve the middle lamellas and allow the cells to separate from each other. Fibers are long and narrow, shorter, wider cells are parenchyma. Scleriids are more or less cuboidal, definitely not long like fibers. These have remained alive at maturity, and nuclei and cytoplasm are visible in several. Blue-stained channels that cross the walls are pits with cytoplasm. Pits of each cell connect with those of surrounding cells so that nutrients can be transferred from cell to cell, keeping them alive. This portion of a water lily leaf contains large, irregularly branched cells stained red. These are astroscleriids. The large white spaces are giant intercellular spaces. This is an parenchyma type of parenchyma. A star-shaped scleriid in a water lily leaf shown by scanning electron microscopy. These cells are located in a large intercellular space. Many have additional arms that extend back into other, smaller intercellular spaces. Figure 5:11 wood is composed of several types of cells. The numerous small cells with thick walls and extremely narrow lumens are fibers, which give wood strength and flexibility. The large round cells that appear to be empty are vessel elements, discussed later in this chapter. The small cells with thin walls and large lumens are wood parenchyma cells. Scleriids are short and more or less isodiametric. Because scleriids have strong walls oriented in all three dimensions, sclerenchyma tissue composed of scleriids acts brittle and inflexible. Masses of scleriids form hard, impenetrable surfaces such as shells of walnuts and coconuts or pits or stones of cherries and peaches. Flexibility there would be disadvantageous because the soft seed inside might be crushed even though the shell remained unbroken. This is similar to our fragile brain being protected by our strong, inflexible, elastic skull. When strength or resistance is the only selective advantage of sclerenchyma, the protoplast usually dies after the secondary wall has been deposited. A mature coconut shell is protective whether its cells are alive or dead. But in some species, certain sclerenchyma cells, especially fibers, remain alive at maturity and carry out an active metabolism. These living sclerenchyma cells most often are involved in storing starch or crystals of calcium oxalate. Some have rather thin secondary walls, but in others, the secondary walls are just as thick as those of fibers that die at maturity and provide only support. Like all cells, scleriids and fibers develop from cells produced by cell division. When newly formed, they are small and have only a primary wall. They are parenchyma cells. If the cell is to differentiate into a scleriid, it may expand only slightly, but if it is to develop into a fiber, it elongates greatly. When immature scleriids and fibers reach their final size, cellulose-synthesizing rosettes in the plasma membrane begin to deposit the secondary wall. The secondary wall is located interior to the primary wall. As it becomes thicker, the protoplast must shrink, usually by removing water from its central vacuole. Secondary walls become impregnated with lignin, making them waterproof, so nutrients can enter the cell only through plasmodesmater rather than everywhere, as with parenchyma and cholenchyma cells. It is important that the secondary wall not be deposited along the entire inner surface of the primary wall. Small, plasmodesmater-rich areas must remain free of the secondary wall. At first, these areas are low depressions in the developing secondary wall, but as wall deposition continues, these areas become narrow pits in the secondary wall. The pits of adjacent sclerenchyma cells must meet. Two pits make up a pit pair. If the pits of one cell met areas of secondary wall in the neighboring cells, no water or sugars could be transferred and the protoplasts would starve. Figure 5:12 These fiber cells have nuclei. They are living cells. Their secondary walls are thick, but not so thick as those shown in Figure 5:11. The small dots in the walls are pits. These are much narrower than the pits shown in Figure 5:10. Leaf of smilax. Conducting sclerenchyma transports water and is one of the types of vascular cells. It is discussed later in this chapter. External organization of stems. The terms stem and shoot are sometimes used interchangeably, but technically, the stem is an axis, whereas the shoot is the stem plus any leaves, flowers, or buds that may be present. All flowering plants have the same basic stem organization. There are nodes where leaves are attached, and internodes, the regions between nodes. The stem area just above the point where a leaf attaches is the leaf axil. Within it is an axillary bud, a miniature shoot with a dormant apical meristem and several young leaves. It is either a vegetative bud if it will grow into a branch or a floral bud if it will grow into a flower or group of flowers. The bud is covered by small, corky, waxy bud scales that protect the delicate organs inside. At the extreme tip of each stem is a terminal bud. In winter, when all leaves have abscised, leaf scars occur where leaves were attached. Plants and people. Box 5-1 Parenchyma, Sclerenchyma, and food. Most of the fruits, vegetables, and other plant parts we eat consist of almost pure parenchyma. With their thin walls, parenchyma cells are soft and easy to chew whether fresh or cooked, and they can be ground, mashed, and sliced for processed food. Whereas cholenchyma and sclerenchyma are used for strength, parenchyma cells are the sites for synthesis and storage of an amazing variety of organic compounds, carbohydrates, fats, proteins, vitamins, pigments, flavors, and other nutritious materials essential to our health. Parenchymatous foods are easy to recognize because we can bite through them easily: apples, pears, strawberries, blueberries, potatoes, lettuce, spinach, and so on. All seeds such as popcorn, beans, rice, and wheat are parenchyma as well, but in their dry, ungerminated, uncooked condition they are too hard to be edible, despite having thin cell walls. Boiling allows water to loosen hard starch grains and protein bodies and to soften the walls. Cholenchyma is not present in all plants and is never really abundant. It is almost always just a minor fraction of all the cells in leaves and stems and virtually never occurs in roots. The most familiar cholenchymatous food is celery. We eat the petioles of leaves, and each ridge along the surface is a mass of cholenchyma cells. Each vascular bundle inside the petiole also has a cap of cholenchyma along one side. These bundles cause celery's stringiness. Other leafy vegetables with thick petioles, like rhubarb and bok choy, also have abundant cholenchyma. Most of us eat these for their flavor and nutrients, not for the pleasure of chewing endlessly on cholenchyma strands. Plants can store starch and proteins in fibers, but they do so only rarely. With the low nutrition and difficulty in chewing, we never use truly fibrous material like mature bamboo shoots and wood for food. However, many vascular bundles contain fiber cells, so even when eating parenchymatous foods we still consume some sclerenchyma. The fibers may be very noticeable, as in asparagus that is a bit too old, green beans, snow peas, artichokes, pineapples, and mangoes. In others, the fibers are a bit softer, especially after being cooked, but you may still notice them in things like squash, pumpkin, and zucchini. You have probably noticed when carving jack-o'-lanterns that cleaning a pumpkin is really just a matter of removing seeds and fibers. Scleriids also occur in some of our foods. Clusters of them cause the grittiness in pears. The seed coats of beans, peas, and most other seeds are also made up of scleriids, as is the covering on seeds of corn. The pieces that get stuck in our teeth while eating corn on the cob are composed of scleriids. In small seeds that are ground into flour, the sclereid-rich seed coats are broken down into small pieces that are easy to eat. Scleriids also make up the brown covering of unpopped popcorn, and popping breaks up the covering. In all cases we do not digest any part of the secondary walls of sclerenchyma cells, all the walls simply pass right through us. The arrangement of leaves on the stem, called phyllotaxy, is important in positioning leaves so that they do not shade each other. If only one leaf is present at each node, the stem has alternate phyllotaxy, 2 leaves per node is opposite phyllotaxy, and 3 or more per node is whorled. The orientation of leaves at one node with respect to those at neighboring nodes is also important. In distichous phyllotaxy, leaves are arranged in only 2 rows, as in corn and irises. In decussate phyllotaxy, leaves are arranged in 4 rows. This occurs in only some species with opposite leaves. Finally, in spiral phyllotaxy, each leaf is located slightly to the side of the ones immediately above and below it, and leaves form a spiral up the stem. This is the most common arrangement and may involve alternate, opposite, or whorled leaves. Table 5, for phyllotaxy. Figure 5.13. In these buds of pistachio, leaves are modified into bud scales, just as those of prickly pear buds are modified into spines. Bud scales are waterproof and protective, but they are shed when the bud begins to grow in the spring. There are buds in the axils of the leaves and a bud at the very tip of the shoot. B. The 2 axillary buds of this viburnum have already begun to grow into branches, and their first young, expanding leaves are visible. These buds were formed in the spring and started growing immediately. They did not form dormant buds with bud scales. All flowering plant shoots are based on the simple arrangement of nodes and internodes, and diversity and specialization are variations of this arrangement. In vines, internodes are especially long, whereas in lettuce, cabbage, and onions, internodes are so short that leaves are packed together. Internodes can be wide, intermediate, or narrow. The diversity is not random. Different types provide particular adaptive advantages in certain situations. For example, plants are physically bound to the site where they happen to germinate as a seed, which by accident may be a shady area near a more optimal sunny spot. Vines, with their elongated internodes, are a means by which a plant can explore its immediate surroundings, and shoots that happen to grow into a sunnier site may flourish. In some species of climbing vines, support and attachment are provided by tendrils, modified leaves, or lateral branches capable of twining around small objects. Figure 5.14. As a leaf falls from a stem, it breaks cells and creates a wound. A leaf scar is a layer of cork that seals the wound, keeping fungi and bacteria out and preventing water loss. Figure 5.15. Phyllotaxy. These young leaves of red bud are arranged with alternate phyllotaxy, 1 leaf per node. Each higher leaf is smaller and younger. Leaves of salvia show opposite phyllotaxy, 2 leaves per node, each pair pointing 90 degrees away from previous and subsequent pairs. Irises have distichous phyllotaxy, 1 leaf per node, arranged in just 2 rows. Iris leaves are flattened from side to side, not from top to bottom. Figure 5.16. Cabbage nodes are packed closely together, and all leaves are tightly clustered. Such closely packed, large leaves with spiral phyllotaxy are poor at photosynthesis and did not evolve by natural selection. They were produced by plant geneticists. The capacity to explore is even more advanced in stolons, also called runners. Their internodes are especially long and thin, and their leaves do not expand. Thus, stolons extend greatly without using much of the plant's nutrient reserves. After the stolon encounters a suitable microhabitat, subsequent growth is by shorter, vertical internodes and fully expanded leaves. New roots are established, and the end of the stolon resembles a new plant. If older parts of the plant die, these vertical shoots become independent plants. After they have started growing vigorously, these plants send out stolons of their own and explore further. In some shoots, nutrient storage is particularly important for survival. These shoots are often massive and quite fleshy, thus providing many parenchyma cells in which starch accumulates. Bulbs are short shoots that have thick, fleshy leaves, whereas corms are vertical, thick stems that have thin, papery leaves. There is no obvious selective advantage of one over the other. The type seems to depend on whether mutations affecting the stem or leaf happen to occur first. Rhizomes are fleshy horizontal stems that allow a plant to spread underground. Tubers are horizontal like rhizomes, but they grow for only a short period and are mainly a means of storing nutrients. The word tuber is often used informally for any bulky underground plant organ. Figure 5:17. These grapevines place their leaves in the full sun at the top of the forest canopy, even though they have not invested much sugar in building strong trunks. The pines that support these vines may ultimately die because they are so heavily shaded by the grape leaves. Various vines climb by different methods. This cocculus grows in a spiral pattern and twines around slender supports. Coral vine climbs by means of branches that bend in a zigzag at each node, but then finally produce a tendril that wraps around objects. After the tendril attaches, the joints bend and draw the vine closer to the support. Before the tendrils attached, the jointed branch was almost straight. Figure 5:18. Airplane plant is a popular ornamental plant that spreads by runners in nature. Most people are familiar with it as a hanging basket plant, with its runners arching out and then drooping down and forming new plants at the tips. The new plant here has already produced roots. All of these storage chutes are subterranean, which is more advantageous than an exposed surface location. Only perennial plants that go through a dormant period need storage capacity. Dormancy is the means by which perennial plants of harsh climates survive the stress of winter cold or summer heat and dryness. They often shed their leaves, reducing water loss. In order to produce new twigs, leaves, and roots in the favorable season, they must draw on stored carbohydrates. Protection of their nutrient reserves, corms, bulbs, rhizomes, and tubers is most easily accomplished by burying them at depths that do not freeze or dry out. A subterranean location also hides them from most herbivores. Each of these specialized tasks is accomplished by characteristic modifications in stem structure and metabolism. As with multicellularity, specialization is accompanied by division of labor, modifications that increase a stem's ability to survive, spread, or store nutrients decrease its efficiency at other tasks. No plant consists only of rhizomes, which are not exposed to light, because most plants must photosynthesize. At least some of the rhizome's axillary buds must grow upward above ground and have green leaves. Similarly, a plant cannot be made up only of stolons. Modifications that allow quick, low-cost exploration are not appropriate for growth, photosynthesis, and sexual reproduction. Another example are the cactus-like spurges of African deserts. Each has a thick, succulent green shoot that photosynthesizes, stores water, and grows longer each year, whereas its axillary buds grow out as slender, short red branches that become fibrous then die and function as spines. It is common for individual plants to have several types of stems and leaves, each of which contributes uniquely to the plant's survival. Although the axil of every leaf contains a bud, only a few buds ever develop into a branch, the others remain dormant or produce flowers. Axillary buds do not become active at random. Each species has a particular pattern. In shady environments, it may be advantageous for axillary buds to remain dormant so that all resources are concentrated in the growth of the vertical main shoot, the trunk, allowing the plant to reach brighter light in the top of the forest canopy. In an open environment, such dominant upward growth is not particularly advantageous. It may be better for all buds to grow and thereby maximize the rate at which new leaves are formed. On almost all plants, at least a few axillary buds remain dormant and serve as reserve growth centers. As long as the apical meristem is healthy and growing well, some axillary buds are unneeded. If the apical meristem is killed by frost, insects, or pruning, axillary buds become active and replace it, allowing the growth of the shoot to continue. Figure 5:19. Onions are bulbs. They have short vertical stems and fleshy leaves. Gladioluses have corms with fleshy stems and papery leaves. A tuber is a short horizontal stem that grows only for a limited period of time. Its leaves are microscopic, and its axillary buds are the potato's eyes, shown here by light microscopy. Figure 5:20. Division of labor. False Solomon's seal consists of subterranean rhizomes and aerial shoots. This succulent spurge has two types of shoots, the long wide green shoot that lives for many years, and many narrow, red shoots that die quickly then function as spines. Figure 5.21 In a heavily shaded forest, many axillary buds remain permanently dormant, allowing all resources to be concentrated on placing one shoot up into the light. With abundant light, one main trunk is not so advantageous. Instead, many axillary buds grow as branches, allowing rapid production of leaves. Internal organization of stems, arrangement of primary tissues. In order to function properly, a tissue must contain the right cells in the proper arrangement. The same principle is true on a larger scale. In order to function properly, the tissues of an organ must be arranged correctly. Epidermis. The outermost surface of an herbaceous stem is the epidermis, a single layer of living parenchyma cells. This differs greatly from our human epidermis, which is much thicker, consisting of many layers of dead cells. Also, whereas we humans have lungs and a digestive tract that absorbs certain things and expels others, all interchange of material between a plant and its environment occurs by means of its epidermis. Because air is almost always drier than living cells, even when relative humidity is high, preventing loss of water to air is a critical function for the epidermis of land plants. Also, the epidermis is a barrier against invasion by bacteria and fungi as well as small insects. It shields delicate internal cells from abrasion by dust particles, passing animals, or leaves, and stems that might rub together, and its reflectivity protects the plant from overheating in bright sunlight. Although it might seem that thick-walled sclerenchyma cells would be more protective, epidermis cells are almost always thin-walled parenchyma cells. Plants do things differently. Box 5 to 2 organs, replace them or reuse them. Plants have 3 types of organs: roots, stems, and leaves. Animals have many types: heart, brain, liver, eyes, kidneys, urinary bladder, and so on. Notice that we have only 1 or 2 of each: 1 heart, 1 brain, 2 eyes. Plants, on the other hand, typically have many of each: dozens of stems, hundreds of roots, and thousands of leaves. Also, notice how long each lasts relative to the life of the organism. We have only 1 heart during our entire lifetime. If it fails, we die. Considering all the mechanical work our heart must do, it is remarkable that we do not periodically grow a new one. We have 2 kidneys, so if one stops functioning, we have a backup organ. However, our bodies will not grow a replacement, even if we lose both kidneys. If we are lucky enough to die of old age, the organs we have at that time are the same ones we were born with. Plants are very different. Their biology is based on using an organ just once, then throwing it away and replacing it with a new version. A tree might have thousands of leaves, but it will discard those in autumn and make an entire new set the following spring. When the tree blooms, it might have thousands of flowers, each with many petals and other parts. The petals function for just a day or 2, then wither and fall off. If the flower was pollinated, it will form a fruit, and then that too will fall off. After the flowering period is finished, the plant will have no sex organs whatsoever until the following year, when it makes a whole new set. Think about how different our biology is from that. Our first thought is probably relief that our sex organs do not fall off after use, even if we could grow another set next year. What are the consequences of the plant type alternative of depending on one-use organs versus the animal type of reusable organs? Leaves must be exposed to sunlight if photosynthesis is to occur, so they suffer extensive exposure to ultraviolet light. Ultraviolet light causes mutations in both plants and animals, leading to skin cancer in us and to damaged, non-functional leaves in plants. We avoid ultraviolet light by wearing clothing, or by performing our activities in shade or indoors. But leaves must be in light. In addition, leaves become covered with dust, spores, fungi, bacteria, insect poop, and other debris. They become just as dirty as any animal, but leaves cannot groom themselves. By upsizing their leaves and growing a new set, they get rid of dirty, diseased, broken down organs and start off a new year with fresh, clean ones. If plants had to rely on the same leaves for their entire lifetime, no tree would ever make it to 100 years old. The same principle applies to other one-use plant organs. Some trees live to be not just hundreds of years old but thousands. Even at such an amazing age, whenever the tree undergoes sexual reproduction, it makes brand new flowers, and those are as fresh and vigorous as when the tree was only a few years old. We animals are not so lucky. When we get on in years, we may still be willing, but our reproductive organs are old. In men, sperm cells are produced by division of the same set of cells that became active when we went through puberty, and there is no reason to think that those cells will remain in better shape than those of our skin, eyes, hair follicles, teeth. In women, cells that will mature into eggs all begin meiosis before we are even born and have been in cell cycle arrest ever since, with just one egg maturing each month. By the time we are 40 or 50 years old, so are all our potential egg cells, but not in plants, not even in old plants. All of their eggs and sperm cells are brand new maiden flowers that have just been produced only a week or two before. Even our circulatory systems follow this difference. We have just one set of blood vessels all our lives. At this point, your aorta is about 20 years old, as are the arteries that take blood up to your brain. All the blood that goes to and from your lungs is passing through veins about 1/5 of a century old, and they are getting older as you read this. Not in plants, however. All woody plants make an entirely new vascular system every year. Every springtime, plants make a new layer of wood that conducts water and a new layer of bark that conducts sugars and minerals. The wood and bark from last year may still be functional, or it may be damaged by fungus, however, that does not matter. There is a set of new tissue, as healthy and functional as our human tissues were when we were babies. Plants and animals are not totally different, actually. We threw away our baby teeth and replaced them with a set of permanent teeth when we were about 6 years old, a very plant-like thing to do. And sharks do not stop with just 2 sets, they constantly produce replacement teeth as long as they live. Birds replace their feathers every year, and starfish grow a new arm if one is cut off. Some lizards grow a new tail, but such capacity does not occur in us. Other than baby teeth, our organs must last us a lifetime. But any time you look at a plant, you are seeing only a tiny fraction of the plant. You are not seeing all the parts that have already existed and fallen off, nor all the parts that the plant will have in the future. If all those parts could be gathered up and preserved, the total plant would be vastly greater than it ever is at any particular time. Figure 5.22. The cuticle on this ivy has been stained dark pink. The lighter layer is wall material impregnated with cutin. Epidermal protoplasts have stained dark blue. Interior cells are photosynthetic parenchyma. 120. This epidermis was taken from a desert plant, agave. Its cuticle is much thicker than that of ivy, and the outer walls of the epidermal cells are extremely thick. Both the wall and cuticle strengthen the leaf surface against insects and prevent water loss. The outer tangential walls are encrusted with cutin, a fatty substance that makes the wall impermeable to water. In species that occur in mild, moist habitats, such incrustation may be sufficient for water retention, but in most plants, cutin builds up as a more or less pure layer called the cuticle. Under more severe conditions, even a cuticle is not sufficient, and a layer of wax may be present outside the cuticle, figure 5.23 in table 5. Cutin and wax resist digestive enzymes and provide defense against pathogens like fungi and bacteria. In some species, the cuticle is so smooth that fungal spores cannot even stick to it and are washed off by rain or shaken off by wind. Waxes are indigestible and non-nutritious, therefore, a thick layer of wax makes it difficult and unrewarding for an insect to chew into a stem. Figure 5.23 On this epidermis, wax is present as flat plates, but in other species it can occur as threads, beads, flakes, or liquid. Unfortunately, cutin and wax also inhibit the entry of carbon dioxide needed for photosynthesis. A totally impermeable epidermis would lead to a plant's starvation. However, the epidermis contains pairs of cells with a hole between them. Guard cells and a stomatal pore together constitute a stoma. Stomatal pores can be opened during the daytime, permitting carbon dioxide to enter the plant. The opening of the stomatal pore is made possible by the structure of the guard cells. Guard cell walls have a special radial arrangement of cellulose microfibrils, which causes some parts to be weaker and more extensible than others. Guard cells swell by absorbing water. The walls next to the stomatal pore are rigid and do not extend, but the back walls are weak and stretch, causing the cells to form an arc and push apart, opening the pore and allowing entry of carbon dioxide and exit of oxygen. This unavoidably allows water vapor to escape and also increases the risk of microbes entering the plant. After sunset, when photosynthesis is impossible, it is not advantageous to keep stomata open. Guard cells in most species shrink and straighten, closing the pore and preventing water loss. Table 5. 5 special chemicals in walls. Figure 5.24 Light microscope view of the epidermis of Zebrina. The stomatal pore, guard cells, and adjacent cells are visible, as are ordinary epidermal cells. Scanning electron micrograph of the epidermis of a cactus showing numerous stomata. Notice how close together stomata are. No chlorenchyma cell inside the cactus is far from a point where carbon dioxide can enter. The stomatal pore is surrounded by guard cells and then other epidermal cells. Notice the small particle of debris at one end of the pore. Clogging by debris is a problem in dusty areas. In most plants, some epidermal cells elongate outward and become trichomes, also called hairs. Trichomes make it difficult for an animal to land on, walk on, or chew into a leaf. They shade underlying tissues by blocking some incoming sunlight, which may be too intense in summer or in high alpine environments. Trichomes can also create a layer of immobile air next to a leaf surface, which allows water molecules that diffuse out of a stoma to bounce back in rather than be swept away by air currents. Figure 5.25 When guard cells absorb water and swell, they become curved and the inner walls are pulled apart by the expansion of the back walls. If guard cells lose water and shrink, they straighten, closing the stomatal pore. Trichomes exist in hundreds of sizes and shapes. Many are unicellular, being just long, narrow epidermal cells. Multicellular ones may be a single row of cells or several cells wide, and many branch in elaborate patterns. Most trichomes die shortly after maturity, and their cell walls provide protection, but others remain alive and act as small secretory glands. Some secrete excess salt, others produce anti-herbivore compounds, and those in carnivorous plants secrete digestive enzymes onto trapped insects. The poisonous, irritating compounds of stinging nettle are held in trichomes. Cortex. Interior to the epidermis is the cortex. In many plants, it is quite simple and homogeneous, composed of photosynthetic parenchyma and sometimes cholenchyma. In other species, it can be very complex, containing many specialized cells that secrete latex, mucilage, or pitch. Some cortex cells contain large crystals of calcium oxalate or deposits of silica. Cortex cells of most plants fit together compactly, but in fleshy stems such as tubers, corms, and succulents, cortex parenchyma is aerenchyma, an open tissue with large intercellular air spaces. A few angiosperms, such as water lilies and the plants grown in aquaria, have become aquatic, living submerged in lakes or oceans. These plants have large cortical air chambers that provide buoyancy. The stems have such a strong tendency to float that no sclerenchyma is necessary for support. Figure 5.26 Trichomes. Flat, scale-shaped trichome. Branched trichomes. Glandular trichomes, these are two large single cells. Glandular trichome with a stalk cell elevating the secretory head cell. Glandular trichome, capable of injecting poison. Simple, unbranched, non-glandular trichomes. Figure 5.27. The cortex of the stem of buttercup is the narrow band of cells between the epidermis and the vascular bundles. The cortex of this corn stem is even narrower, in some places only 2 or 3 cells wide. Vascular tissues. For very small organisms whose bodies are either unicellular or just thin sheets of cells, diffusion is adequate for the distribution of sugars, minerals, oxygen, and carbon dioxide throughout the body. But if any cells of the organism are separated from environmental nutrients and oxygen by just 4 or 5 cell layers, diffusion is too slow, and a vascular system is necessary. 2 types of vascular tissues occur in plants: xylem, which conducts water and minerals, and phloem, which distributes sugars and minerals. Vascular systems of plants are quite different from those of animals. The plant vascular system is not a circulatory system. Water and minerals enter xylem in the roots and are conducted upward to leaves and stems. During its passage, xylem sap travels through dead, hollow cells, not through tubes composed of living cells like our blood vessels. Once in the shoots, most water evaporates from the surfaces of stems, leaves, and flowers and is lost. The minerals and a bit of water are used by surrounding cells. Phloem cells are living. They pick up sugar from areas where it is abundant, usually leaves during summer and tubers or rhizomes in spring, and transport it to areas where sugar is needed, especially growing tips of shoots, roots, young leaves, and flowers. In the later months of summer, phloem carries sugar into developing fruits and into the storage organs of perennial plants. Because sugar must be dissolved to be conducted, water is transported simultaneously in phloem. Xylem. Within xylem are 2 types of conducting cells, tracheids and vessel elements. Both are types of sclerenchyma. The term tracheary element refers to either type of cell. As a young cell matures into a tracheary element, it first must enter cell cycle arrest and stop dividing. It is initially a small parenchyma cell with only a thin primary wall, but the cell becomes long and narrow and then deposits a secondary wall that reinforces the primary wall. The cell then dies, and its protoplasm degenerates, leaving a hollow tubular wall. Alternatives. Box 5 to 3. Simple plants. 3 groups of plants have much simpler bodies than those described in this chapter. Hornworts are a rather rare group of tiny plants whose bodies look like bits of green cellophane, often no larger than 2 to 5 millimeters in diameter and only a few cells thick. They have no epidermis, no stomata, no parenchyma, no sclerenchyma, no cortex, no pith, no xylem, and no phloem. Instead, they are small ribbon or disc-shaped sheets of parenchyma cells, most of which have a single chloroplast. Each cell absorbs water and minerals, carries out its own photosynthesis, and is relatively self-reliant. When the plant reproduces, horn-like structures grow upward, break open, and release spores. During this stage, nutrients are transported from chlorophyllous cells to spore-producing cells, but only by means of plasmodesmata, not phloem. Many liverworts are about as simple as hornworts. Plants of Palavicinia, Sphaerocarpos, and Riccia also consist of just small green ribbons or discs of chlorophyllous cells. Bodies of Fossambronia and Petalophyllum appear leafy and more complex, but in fact, they are just ribbons whose edges are puckered. The bit of complexity present in these bodies is merely that the central portion of the ribbon is several cells thick, whereas the margins are unistratose. As in hornworts, there is no epidermis or vascular tissue. Other liverworts such as Asterella and Marcantia have thicker, more complex bodies. The ventral side is several layers of compact parenchyma with few chloroplasts, and the dorsal side consists of intersecting sheets of cells that form air chambers. The term epidermis is used for surface cells, Dorsal epidermis has stoma-like air pores. Ventral epidermis has trichomes that attach the body to soil or tree bark. The most complex liverworts have leaves and are called leafy liverworts, but their bodies are composed of just parenchyma. As in hornworts, liverworts transfer nutrients through plasmodesmata from chlorophyllous cells to spore-producing tissues when they reproduce. Liverworts have no vascular tissues. Despite their simplicity, all liverworts grow by means of organized apical meristems. Mosses are a bit more complex than either hornworts or liverworts. Mosses always have a stem with leaves, and in those with taller stems or larger leaves, some cells deposit a thicker wall and resemble sclerenchyma fibers. However, these walls do not contain lignin and probably have a separate evolutionary origin, so they are called steroids instead of fibers to avoid confusion. Many mosses conduct water, but along the outside of their bodies, not inside and not with xylem. Their numerous leaves are so tiny and close to the stems they form capillary spaces and act like a wick. A number of mosses do have an internal system of conducting cells in their green, leafy stems. Cells called hydroids conduct water, and others called leptoids conduct carbohydrates and other nutrients. These should never be called xylem or phloem because they differ in significant ways from those two tissues. What are some consequences of having such simple bodies? The simplicity of hornworts, liverworts, and some mosses limits their ability to survive in some habitats. Lacking conducting tissues, they must remain short, thin, or prostrate, and many quickly die in dry air. On the other hand, the same simplicity enables them to survive in many habitats because just a few weeks of cool moist weather is enough to allow their spores to germinate and grow into tiny, mature plants. They do not need to photosynthesize long enough to produce all the carbohydrates that a vascular plant needs for its larger, more complex body. Even deserts have rainy periods and shaded spots that stay moist for weeks. Such spots will almost always have some non-vascular plants thriving there. Figure B5, 3a plants of hornworts. The bodies are just thin, green sheets of parenchyma. The columns are reproductive structures. Figure B5, 3b plants of this liverwort grow in quiet streams. Each shoot is less than 1 mm thick and consists of parenchyma cells. Each grows from an apical meristem that occasionally divides in two. Figure 5, 28 tracheary elements. Tracheids are long cells with tapered ends. The primary wall is complete over the whole surface, and it is so thin that the inner, spiral secondary wall is visible. The primary wall tore during specimen preparation, revealing just how thin it is and permitting views of the circular bands of secondary wall which are located just interior to the primary wall and which prevent it from collapsing during water conduction. Vessel elements tend to be wider and shorter than tracheids, but the most important feature is the perforation, the large hole at each end. The perforation of one vessel element must be aligned with that of the next if water is to pass through with little friction. The stack of vessel elements is a vessel. The vessel here has been cut down the center, so the front wall and back wall are missing and the vessel appears to be just an open space. 600,270,100. 1,250. Table 5, 6 tracheary elements. The secondary wall is impermeable to water, thus areas of the permeable primary wall must remain uncovered if water is to enter and leave the cell. In the simplest type of tracheary element, there is only a small amount of secondary wall, organized as a set of rings, called annular thickenings, on the interior face of the primary wall. This arrangement provides a large surface area for water movement into and out of the cell, but it does not provide much strength. The primary wall must be supported because movement of the water tends to cause it to collapse inward. With helical thickening, the secondary wall exists as 1 to 3 helices interior to the primary wall. Scalariform thickening provides much more strength because the secondary wall underlies most of the inner surface of the primary wall and is fairly extensive. Just as in fibers and sclerids, the area where the secondary wall is absent is called a pit. In tracheary elements with reticulate thickening, the secondary wall is deposited in the shape of a net, as the name suggests. The most derived and strongest tracheary elements are those with circular bordered pits. In such tracheary elements, virtually all of the primary wall is underlain by secondary wall. The pits that allow water movement are weak points in the wall, but the weakness is reduced by a border of extra wall material around the pit, hence the name. Tracheary elements with annular thickenings are weak, but a large percentage of their primary wall is free of secondary wall and available for water movement. Pitted tracheary elements are just the opposite. They are extremely strong, but so much of the surface is underlain by lignified secondary wall that water enters or leaves the cells slowly. Each is selectively advantageous under certain conditions and disadvantageous under others. If a species grows in perpetually wet soil and water moves easily, its cells are not in danger of collapsing, and thus, tracheary elements with annular or helical secondary walls are adaptive, not only because the large open regions of primary wall allow water to pass through quickly but also because the plant uses little sugar-building secondary walls. If soil dries even slightly, however, water is pulled with more force and exerts inward traction on tracheary element walls. The stronger scleriform, reticulate, or circular bordered pitted walls are necessary to keep the tracheary elements open. Although they cannot conduct water as quickly, water movement is automatically slower because the soil is dry. Under dry conditions, using extra glucose to build stronger secondary walls can be a valuable investment. Figure 5, 29 tracheids. The growth of a young cytoplasmic cell into a tracheid. After the cell has almost reached mature size, the secondary wall is deposited interior to the primary wall. After the secondary wall is finished, the protoplasm dies and degenerates, leaving only the primary and secondary walls. A tracheid cut open, showing a secondary wall in the form of helical thickenings. A whole cell in which the primary wall is so thin that the annular secondary walls inside are visible. Whole cell with helical secondary wall. Scleriform secondary wall. Reticulate secondary wall. Pitted secondary wall. Light micrograph of a tracheid with a helical secondary wall. Figure 5:30. As water enters a tracheary element, it passes through primary wall between regions of secondary wall. Water adheres to cellulose molecules, pulling walls inward and upward as it rises in xylem. Primary walls tend to collapse, but secondary walls provide strength to hold the primary wall in place. Under severe water stress, annular and helical secondary walls may not be strong enough to prevent collapse of the primary wall. Tracheids obtain water from other tracheids below them and pass it on to those above. They must occur in groups, lying side by side with some and having their ends overlapping the ends of others. Also, the pits of adjacent tracheids are aligned so that water can pass through. The aligned set of pits is a pit pair, and the set of primary walls and middle lamella between them constitutes a pit membrane, just as in fibers and sclerids. Although very permeable to water, a pit membrane, being a set of primary walls, does offer slight resistance. If each tracheid is 1 mm long, each water molecule passing from root to leaf in even a short plant only 1 meter tall must pass through 1,000 pit membranes. The friction adds up. Figure 5:31. Side view of a pit that has been bisected. In a sclerid or fiber, the secondary wall would be thicker and the pit narrower and deeper. The diameter of the pit can vary, being broad as in figure 5:10d or narrow as in figure 5:12. A bordered pit has a rim of extra wall material that prevents the pit from being a weak spot in the wall. With borders, pits can be broad enough to allow rapid flow of water from tracheary element to tracheary element, but not so weak as to be detrimental. Only tracheary elements have bordered pits, fibers and sclerids do not. Figure 5:32. Bordered pits in face view on front wall of a vessel element. The white slits are the actual openings of the pits, the pinkish area around each white region is the border, and the dark red hexagons are regions where the secondary wall lies against the primary wall. Bordered pits in face view on the front walls of tracheids of pine. This is the same magnification as, notice that tracheids are much narrower than vessel elements, and pits in pine are extremely large. Scalariform bordered pits. Vessel elements provide a way to move water with less friction than tracheids. Vessel elements, like tracheids, are individual cells that produce both primary and secondary walls before they die at maturity. In vessel elements, however, an entire region of both primary and secondary wall is missing. During the final stages of differentiation, a large hole called a perforation is digested through a particular site of the primary wall, often removing the entire end wall.